Hi, welcome back to Chapter 7. In the second video, we'll cover lipids, proteins, and how biochemistry helps identify microorganisms. By the end of this section, you should be able to describe the chemical composition of lipids, explain their unique characteristics and structure, compare triglycerides and phospholipids, and describe how phospholipids form membranes. Lipids are mostly made of carbon and hydrogen, but sometimes also with oxygen, nitrogen, sulfur, or phosphorus. They act as nutrients, store energy, build membranes, or function as hormones and pigments. A key property is that they are either hydrophobic or nonpolar, meaning they don't mix with water, or amphipathic, which means they have both hydrophobic, water-fearing, and hydrophilic, water-loving parts. This makes them crucial for building cell membranes. If you need a review of vocab terms like nonpolar or just a general chemistry review, please see Appendix A for more information. Fatty acids are long hydrocarbon chains with a carboxyl group. If they are saturated, so if we have saturated fatty acids, these have no double bonds in their fatty acid tail, meaning they're straight in their structure, and these fatty acid tails without bends, if they are straight, then they pack tightly, so these saturated fats tend to be solid at room temperature like butter. Unsaturated fatty acids, on the other hand, have at least one or more double bonds in their fatty acid tails, making them bent in structure so they cannot pack tightly, and they tend to be liquid at room temperature like oils. Triglycerides, also known as triacylglycerols, are composed of 3 fatty acids linked to a glycerol molecule, and these are the main form of energy storage in adipose tissue, and they also make up sebum. We see the formation of triglycerides here. There is a glycerol molecule on the left, 3 fatty acid tails here on the right, and when they're put together through the reaction dehydration synthesis, we form the triglyceride and 3 water molecules. Our first checkpoint asks why fatty acids with hydrocarbon chains that contain only single bonds are called saturated fats. So these are because if we look at just the fatty acid tail, remember that when we're talking about those terms, saturated versus unsaturated, we're not looking at the head, just at the tail portion. So in saturated fatty acids, each carbon is bonded to the maximum number of hydrogen atoms possible. There are no double bonds present, so the chain is saturated with hydrogen. It's full of hydrogen. This gives the chain a straight, flexible shape and makes it solid at room temperature because other fatty acid tails could pack tightly against this one. In contrast, unsaturated fatty acids have one or more double bonds in their structure in that fatty acid tail, giving them a bent shape, which makes it hard to tightly pack other fatty acid tails against this one. And these tend to be oils. These tend to be oils at room temperature. There's one I didn't talk about yet, known as trans fats. Trans fats are actually also unsaturated, but they behave like saturated fats, right? They look more like saturated fats because they have again, this kind of straight chain. And trans fats do exist in nature, but in small amounts. Most trans fats are man-made. Phospholipids look like triglycerides, but here one fatty acid is replaced with a phosphate group. So here is my glycerol, and usually I have 3 fatty acids, right, in triglycerides, but in phospholipids this one is not there. Instead, what's attached to that last carbon in my triglycerol molecule is a phosphate group. Because of this gives them a polar, water-loving head or hydrophilic head for these phospholipids, and 2 nonpolar tails. These are hydrophobic tails. So this makes phospholipids amphipathic. They have a water-loving head and water-fearing tails. Because of this characteristic, being amphipathic and partly water-loving, partly water-fearing, phospholipids can self-assemble into bilayers or micelles or liposomes in water. And this property is the foundation of biological membranes. So in a regular cell, the cell membrane is made up of a phospholipid bilayer, kind of like the one shown here. And if this were a cell, I think the central compartment would be larger. But in here, the central compartment, we would have like our organelles, our nucleus, ribosomes, etc. And the membrane is a double bilayer. And I'm going to zoom in on just this section over here, where I can see the polar water-loving heads are facing outside of the cell, but also facing inside of the cell. And my lipid nonpolar water-fearing tails are facing each other in the central portion. Because of their structure, these phospholipids can also form single-layer lipid spheres where the polar heads are facing outwards and all the nonpolar water-fearing tails are facing inwards. These micelles in the middle always remind me of soap, how soap works. So when you're washing your hands with soap, the soap particles reform into these micelle structures or micellar structures where oil and dirt is trapped in the middle, where the phospholipid tails are, and the water-loving heads are on the outside so that they can be washed away with water. Micellar water, you might have seen that in some kinds of like makeup remover, is basically just soap water. This checkpoint asks, how is the amphipathic nature of phospholipids significant? Remember, phospholipids have both hydrophilic heads and hydrophobic tails. So in water, they can self-assemble into structures like micelles, these bilayers where the tails are facing each other, or liposomes, a lipid bilayer that is in a spherical form. And this property forms the foundation of cell membranes, creating barriers and compartments essential for life. Another lipid type are isoprenoids, also called terpenoids. These are branched lipids that are found in pigments, vitamins, fragrances, and waxes. These are formed from chemical modifications of the isoprene molecule. Sterols are ring-shaped lipids like the ones shown below here. Examples include cholesterol in animal cell membranes, ergosterol that can be in fungal or protozoa membranes, and hopanoids in bacteria that might be in their membranes. These sterols give cell membranes strength and stability. So how are isoprenoids used in technology? Isoprenoids or terpenoids are chemically modified isoprene molecules, and we see them in our daily lives, but although you might not have heard of the term isoprenoids before. So applications include in pharmaceuticals, forming capsaicin in pain relief creams, forming pigments like beta-carotene or xanthophylls, in fragrances like menthol or camphor or limonene, and in waxes and oils like water-resistant coatings or skin protection. All right, let's recap lipids. For the first one, chemical composition. They're made of carbon and hydrogen, but can also contain oxygen, nitrogen, sulfur, or phosphorus. They can be completely hydrophobic or nonpolar, or amphipathic, where they have both water-loving and water-fearing regions. For the second one, unique characteristics. Again, they're hydrophobic or amphipathic, which influences their solubility and membrane formation. They're highly diverse, so we saw fatty acids could be saturated and straight and solid, or unsaturated, being bent and liquid. We have triglycerides for energy storage, phospholipids for our membranes, and isoprenoids, or also known as terpenoids, for pigments, fragrances, vitamins, or waxes. And we have sterols, like cholesterol, ergosterol, or hopanoids to strengthen membranes. For the third one, triacylglycerides or triglycerides. Remember, these are made of 3 fatty acids connected to a glycerol molecule. They're nonpolar or hydrophobic, and they function in energy storage in adipose tissue or in oils or in sebum. For phospholipids, we have 2 fatty acids with a glycerol plus a phosphate group. That makes them amphipathic, so they have a water-loving polar head and 2 nonpolar tails, and their function is in membrane structure. For the last one, how phospholipids are used to construct biological membranes. Because of their amphipathic property, they can self-assemble in water, where hydrophilic, water-loving heads are going to face the water, and hydrophobic tails are going to cluster away from the water. This can allow them to form bilayers or micelles or those liposomes. And bilayers are the ones that are the foundations of cell membranes, which create selective barriers and compartments within our cells. All right, our next section is proteins, 7.4. And by the end of this section, you should be able to describe the structure of amino acids, explain protein structure levels, and summarize why proteins are unique. Proteins are built from amino acids, which contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. They act as enzymes, receptors, hormones, transporters, and structural components, and their diversity comes from differences in the amino acid R groups and the way they fold. So the R in R group stands for residue. Sometimes it's just thought of as the rest of the molecule. And in organic chemistry and biochemistry, R is used as a placeholder for any group of atoms attached to the core structure. So for amino acids, which apply to proteins, the R group is the variable side chain that's bonded to the central carbon, or sometimes it's known as the alpha carbon. The side chain will determine the amino acid's properties, like if it's polar, nonpolar, acidic, basic, etc. Each amino acid has a central carbon, sometimes known as the alpha carbon, with an amino group attached, a carboxyl group attached, a hydrogen atom, and a variable R group, that side chain. Amino acids link together through the reaction dehydration synthesis, forming peptide bonds between each amino acid. So here I can see peptide bonds where these vertical lines are. So these are composed, this chain is composed of 1, 2, 3, 4 amino acids. And if you have a chain under 50 amino acids, we call those peptides. Longer ones are known as polypeptides or proteins. Here's just an example of a few amino acids and their general structure. For all of them, we can see a central carbon and those 4 attachments: the carboxyl group, the amino group, the hydrogen, and the R group. So the R group is highlighted in blue in all of these images, and you can see that the R group will give them their properties, like being polar or water-loving, or nonpolar or water-fearing. The length, or how many amino acids you have in your chain, contributes or impacts the name. Just like we saw for monosaccharides, disaccharides, oligosaccharides, etc., we see that for amino acid chains as well. So if we have 2 amino acids linked together, for instance, we can call that a dipeptide. If we had 3, amino acid 1, amino acid 2, amino acid 3, that would be a tripeptide. If you have a few, those are known as oligopeptides. If you have many, we call those polypeptides. And again, here we see dehydration synthesis, linking 2 separate amino acids together, forming that peptide bond, and water comes out of the reaction. So how many amino acids are in polypeptides? So remember, a polypeptide is just a linear chain of amino acids linked by peptide bonds. The chains we saw can be short, just with a few amino acids, or very long, hundreds or even thousands of amino acids. So for a distinction, we use oligopeptide for short chains, like fewer than 20 amino acids, and polypeptides are for long chains, which can be dozens to even hundreds of amino acids. When we use the term protein, this is going to be 1 or more polypeptides together, folded into some kind of functional 3-dimensional shape. The size or length and specific amino acid sequence of a protein are major determinants of its shape, and the shape of a protein is critical to its function. So if you don't have the right shape for a protein, the substrate won't be able to bind and generate the reaction that is supposed to happen. There are 4 levels of protein structure. It starts from primary structure all the way to quaternary structure over here. Primary structure is the amino acid sequence, so the order of amino acids is important to its final structure. Secondary structure is the local folding of the amino acid sequence, and it'll form structures like alpha helices or beta pleated sheets because of hydrogen bonds, and I'll show you a picture soon. Tertiary structure is 3-dimensional folding stabilized by bonds such as disulfide bonds, ionic bonds, or hydrophobic interactions, and this is due to their R group interactions between the amino acids. And quaternary structure is when more than one polypeptide comes together. So you have multiple subunits or multiple polypeptides interacting to form your final 3-dimensional structure. If a protein loses its shape, we call that denaturation, and the loss of shape means it usually loses its function. Sometimes denaturation is reversible if the primary sequence is still intact. Sometimes it is not reversible. So again, primary structure is the linear sequence of amino acids, and the order of amino acids is important. And notice that they're connected by peptide bonds, a type of covalent bond. Secondary structure is due to the formation of hydrogen bonds within the backbone of the polypeptide, and this is not involving R groups. Here we can see a hydrogen bond forming between a carbonyl group and this hydrogen atom attached to the nitrogen. Here we can form either beta-plated sheets or alpha helices within secondary structure. Tertiary structure comes next, and this is when you finally see that three-dimensional folding of your polypeptide chain into some kind of three-dimensional form, and this is going to be due to the interaction of our R groups, our side chains, and you'll see all different types of bond types forming here, including hydrogen bonds between the R groups, covalent bonds like disulfide bonds forming between the R groups, ionic bonds, or even hydrophobic interactions. So talking about protein structure and tertiary structure always reminds me of hair. Hair is made of keratin protein. So imagine you wake up in the morning and your hair is kind of messy. How do you fix it? Think of things you can do to temporarily change the shape of your hair. You can use heat, apply water, use a blow dryer or a flat iron. These things will disrupt the weaker bonds and reform them in the shape of the hair that you want. But if you want changes that are permanent, like getting a perm to create curly hair like the ones shown here, then you need to form disulfide bridges. These are covalent bonds, so you would have to get some kind of chemical treatment to form covalent bonds. Alternatively, if you have curly hair and you want to have straight hair, you would also have to have chemical treatment to get rid of these covalent bonds. So weaker bonds we can alter with heat or water, but stronger bonds like the covalent bonds would have to be chemically altered. Finally, quaternary structure is when you have more than one primary structure linked together. So basically more than one polypeptide chain interacting together, and you can see all bond types here as well. Ionic, hydrogen, covalent, hydrophobic interactions, etc. So it's not always that it's made of four. So quaternary structure does not necessarily mean there are four polypeptide chains interacting, like we see in hemoglobin, a very common example. You could just have two chains interacting, or three, or even maybe 50. And here's a nice summary of those four levels. In primary structure, this is our linear sequence of amino acids formed by peptide bonds, a type of covalent bond. In secondary structure, we have hydrogen bonds forming alpha helices or beta sheets. In tertiary structure, we have a three-dimensional folding due to all bond types, and not all proteins have quaternary structure. This happens when you have more than one amino acid chain interacting with each other. Proteins are also interesting in that they don't follow the usual pattern in their states of matter. So imagine if you're heating something up. Usually things go from solid to liquid, right? Like a block of ice. I'm going to heat that up. It's going to turn to water. It's going to melt. Same thing for chocolate. I heat that up. It goes from solid chocolate bar to liquid, right? Chocolate goo. For eggs, which are made of protein, I am starting with a liquid. So in this particular case, the egg is liquid at room temperature, the raw egg, and when I cook it, it becomes solid. It goes opposite of what I'm used to. And this is because of protein, right? We have egg albumin, the egg white. When you heat it up, the protein will denature. Water actually can come out of the shell. It's not waterproof, and it becomes solid. So what can happen if a protein's primary, secondary, tertiary, or quaternary structure is changed? Altering structure will change the protein's shape, and losing their shape is known as denaturation, and the protein might no longer function. So for example, if it's an enzyme, it might lose their active site. It won't be able to bind to the substrate anymore. If it's a structural protein, they might lose their stability. And even what seems like a small change, like changing maybe just one amino acid in a long sequence of amino acids, can cause diseases like cystic fibrosis. Cystic fibrosis is caused by a mutation in a membrane transport protein called CFTR. This actually stands for cystic fibrosis transmembrane conductance regulator. Losing just one amino acid, which is phenylalanine in this case, changes the primary structure. The protein will misfold, and it won't transport salt and water like it usually does. As a result, thick mucus will build up in the lungs and organs, which can trap bacteria very easily. In cystic fibrosis patients, bacteria like Pseudomonas aeruginosa can form these biofilms, which makes infections persistent and can be really hard to treat. So this is a really powerful example of how a small change in protein structure can have huge health consequences. So the normal job of the transporter is to transport chloride ions across the membrane, and by moving chloride, it helps control water movement as well to keep mucus thin and slippery. But for the mutation CFTR or mutated CFTR, it can't properly transport chloride ions. Less water enters these mucus secretions, resulting in this really thick, sticky mucus that can build up and increase likelihood for infection. Okay, let's recap proteins. So the fundamental structure of an amino acid, we have the central alpha carbon bonded to an amino group, NH2, the carboxyl group, COOH, a hydrogen atom, and then the variable side chain, that R group, which determines the chemical properties of the amino acid. For the second one, describe chemical structures of proteins. We said proteins are made of amino acids linked by peptide bonds that are formed by dehydration synthesis. We had primary structure, the linear sequence of amino acids. Secondary structure, those alpha helices and beta pleated sheets formed by hydrogen bonding. We had tertiary structure, the overall three-dimensional folding formed by disulfide bridges, ionic bonds, hydrophobic interactions, hydrogen bonds. And then quaternary structure is when multiple polypeptides are assembled together. For the fourth one, unique characteristics of proteins, we have really enormous diversity due to different amino acid sequences and their folding patterns. They can function as enzymes, receptors, hormones, transporters, structural components, and their shape is really critical. We saw how small changes in shape can alter or destroy the protein function. Sometimes proteins can also be conjugated or bound to non-protein subgroups, and we're going to see that later in the formation of glycoproteins or lipoproteins. In this last section of Chapter 7, we're going to use biochemistry to identify microorganisms. So by the end of this section, we should be able to describe examples of biosynthesis products inside cells that can be used to identify bacteria. Biochemical identification of microbes tells us that in the lab, microbes can be identified based on their unique biochemistry. Analyzing the nutritional and metabolic abilities of the bacteria is really a common approach to determine the genus and species of the bacterium you have. So for example, some bacteria store granules like PHB. PHB stands for poly-beta hydroxybutyrate. Don't worry about that. It's a carbon and energy storage compound. These can be stained to identify certain species of bacteria. Metabolic tests will be important in the lab. It'll be our next project. We can use metabolic tests to see if bacteria, for instance, use certain carbon sources, like whether or not they ferment lactose. Some of these are very detailed and related to chemistry, so you're going to learn more about these in your chemistry class, including Molotov. These basically create protein fingerprints of your unknown sample and compare them to known databases, so other protein fingerprints of bacteria we already have identified, to figure out what you have. Lipids can also be used in Molotov identification, but it's not as common. Phase analysis is also very similar. Again, you're going to learn more about gas chromatography and chemistry, so don't worry too much about it. But the basic idea is we're going to take lipids, alter them to form volatile forms of the lipid, so gas forms of the lipid, and compare these chemicals to a database for what we already have, a bunch of lipid profiles for known bacteria to figure out what bacterium we have. Proteomic analysis compares something similar, again, also chem related. We're looking at peptide patterns. So you're going to use, again, something from chemistry known as high pressure liquid chromatography to look at peptide fragments of your unknown bacterium and compare them with the fragments of known microorganisms to figure out what you have. For serological tests, these detect unique surface carbohydrates, so different carbs that are located on your unknown bacterium to what we know exist on known bacteria. So all of these methods let us pinpoint what microbes we have based on what they make or how they metabolize certain nutrients. So again, a lot of these you're going to learn in future classes, but some examples of biosynthesis products were things like that storage granule with the PHB, the polybeta hydroxybutyrate, which is used for energy storage, and we can use stain these to distinguish certain Pseudomonas species from others, because some accumulate PHB and others do not. We can use pigments. So in the lab soon, we're going to see that certain Pseudomonas bacteria produce fluorescent pigments, but others do not. We can use lipids, looking at their unique fatty acid profiles and compare them to our known databases. We can use proteins through proteomic analysis and the procedures from chemistry like mass spectrometry to identify bacteria by their protein fingerprints. We can use carbohydrates like their unique surface carbohydrates in glycoproteins or part of their cell wall and detect these using serological tests like these antibody-antigen interactions. And that takes us to the end of Chapter 7. In Chapter 8, we're going to start talking about microbial metabolism. Thanks for watching.